This recorded session is dedicated to our module on primary and secondary immune responses. In this session, we will refer to a few narratives that we already covered from the previous lectures, particularly those that touched based on lymphocyte development and differentiation, as well as antigen presentation and processing. So you remember from our last module on MHC and complement proteins, no? We already covered so much about how foreign antigens are processed and presented such that the immune system, especially the adaptive immune system, will be able to develop the necessary effectors, both humoral and cellular, to fight the foreign antigen or the foreign microbe. Let us begin by describing how the body responses when sensitizing agents enter the systemic circulation, the blood. So we divide immune responses to an antigen into two phases, into two waves. So we have the primary wave or the primary response, which happens during the first instance that the antigen is encountered by the body. It can take up to 14 days to resolve, so quite long compared to the secondary wave. And the goal or the end point of the primary response is to generate a set of memory cells that have high specificity for the inducing or the sensitizing antigen. The second wave or the secondary response happens during subsequent encounters with the same antigen. But it is usually more rapid, or definitely we expect it to be more rapid compared to the primary response. And what happens during secondary response is the memory cells produced in the primary wave will be activated. So essentially, the fact that it's more rapid to compared to primary response is attributable to the existing pool of memory cells, both B and T cells. No? We will go back to, or we'll have a review of, the elements of innate and adaptive immune response as we go along with the discussion of the events between or behind primary and secondary immune response. We will cover primary response first kasi the molecular underpinnings of the primary response will make us understand better yung events happening in the secondary response. So you will remember from our discussion about innate and acquired immune responses that aside from or after the breaching of the physical chemical barriers, the skin, mucous membranes, acidity of some parts of the body. Okay, after that is breached, no, we expect that the cellular effectors of immunity will come into play, especially the innate immune cells, the phagocytes that recognize foreign cells, foreign antigens, and carry out direct killing, direct processing of them, as well as natural killer cells capable of recognizing virally infected host cells. You know? And then we expect that after the innate cellular response, acquired or adaptive immune response should develop, and the primary actors are the lymphocytes, no, and both T and both T and B, no, ating mga helper and killer, helper and cytotoxic T lymphocytes, as well as the B lymphocytes are the primary cellular immune effectors under the adaptive immune system. The, we expect that eventually, no, these T and B lymphocytes will develop some form of memory. A subset of them will form some will form a type of memory against the sensitizing agent or against the sensitizing antigen, the foreign microbe. And that memory is characterized by the ability to recognize them. So there are specific receptors that are involved or that are already present, and antibodies specific to the antigen or the foreign material, the foreign cell, are expected to be produced during the course of primary response. But eventually, in the secondary immune response, only the memory cells capable of producing the antibodies are expected to be present. Na eventually, yung antibodies produced against the antigens um, will wane out, no? Kokonte, magde-decrease ng kanilang concentration as the primary response comes to a resolution. No? The specificity of the innate processes or the innate immune effectors, generally they're effective against all types of organisms. So you expect that in the primary response, all the effectors involved will be able to carry out the process of immune clearance na without really depending too much on antibody antigen recognition. So very ano siya, very broad yung mechanisms of action of carried out by or exhibited by our immune cells, no? And in our immune effectors in the innate process, no? Or the innate system. While sa acquired naman, the specificity for the targeted organism or the stimulating organism is expected to be developed, to be fine-tuned. So you expect that in the subsequent cases, mga secondary waves of immune response, no? The body will develop the body will respond faster. Okay, there is memory already, so that it will not take long for the immune system to totally clear the invading pathogen. Okay, now the acquired the innate and the acquired or adaptive immune systems, we can in summary, no, we can say that uh, they form a continuum, no? Where your innate immune responses serving as first line of defense, if will not be enough to really result to immune clearance of the pathogen, will trigger adaptive immune responses. And our adaptive immune responses will, of course, uh, include or cover antigen processing, antigen presentation, and eventually the end goal is for memory cells to be developed. Such memory cells will respond to a second, third, or fourth wave of antigen exposure, provided that the same antigen is present. When I say the same antigen, I refer to the nature of the antigen not totally changed compared to the first instance that it was presented, or that it was that it was the first instance that it was processed by the adaptive immune effectors. Um, yun, sabi ko na, no, the goal of which is to develop memory cells actually relate to how prompt, how immediate the secondary immune responses are compared to the primary immune response. Kasi in secondary immune response, the process of antigen presentation, um, affinity maturation, and what do you call this? Yung ano no, development of specific memory cells, it's ano na siya, hindi na siya it will not start in square one anymore. Okay, it will proceed with the processes as is, because of the fact that you already have a pool of memory T and B cells na meron ng specific receptor, meron ng altered immune what do you call them, IG coding genes, no? Meron ng specific na IG coding genes for the antibody. So you already have your pool of memory lymphocytes that will carry out the process of recognition and antibody secretion para mas mabilis, no, yung immune clearance na tinatawag. So, um, as I mentioned, no, the primary and secondary immune response will have specific differences that we want to cover in this lecture. And before we really talked about the difference between, or before we journey into discussing what happens during the primary response and secondary response, we have to introduce or I have to provide a summary. So, pwede nyo itong balik-balikan as you proceed with our, or as you finish the discussions for both primary and secondary response. Pwede nating balik-balikan itong summary table na ito. So, for the primary response and secondary response, antigen presentation is quite different. In primary response, mainly antigen-presenting cells are non-lymphocytes. Dominant is the dendritic cell. It's a primary antigen-presenting cell for the primary response. While for secondary response, B lymphocytes are increasingly important for antigen presentation, unlike in primary response. No? Also, the required dose of antigen for sensitization in primary response is relatively high, while for secondary response, even small concentrations of the same antigen can already trigger immune responses. Now, for the lag phase or the latent phase, we expect that in primary response, it's going to be longer, 1 to 2 weeks, no, 5 to 10 days, while for secondary response should be less than 5 days, no, 2 to 5 days. The frequency of specific B cells for primary response will be high, attributable to the fact that dito nagka-create talaga ng mga specific na B cells. So, mas marami ang specific B cells during primary response. In secondary response, a decline in the level of specific B cells is expected, but you can also, or we can also expect that eventually, if eventually yung number naman or the nature of the specific B lymphocytes in secondary response, very specific sila for the inducing or sensitizing agent. So we're not really after the number, but more of the specificity, the what do you call this, yung exact na capacity to recognize, very high in B lymphocytes present in secondary response. For the type of antibody produced in primary response, the predominant antibody is IgM, while for secondary, predominant is IgG or IgA. So you expect the first type of antibody produced in any primary response to antigen is the IgM. Tapos mag-wane out yan. IgG and IgG is normally the one that will take over, no? Yung magkakaroon ka ng IgG specific to the inducing antigen. And the affinity of antibodies in cells, if we compare the two waves, in primary response, lower affinity of the cells and the antibodies, while for secondary response, as I've already since I've been repeating myself multiple times, very high yung yung affinity ng immune effectors, both cells and antibodies during the secondary response. Okay? So, now we will discuss first the molecular events in the primary immune response, so we can further appreciate how the secondary immune response is capable of carrying out yung faster rates of immune clearance. The primary immune response is given the term or given the name because of the fact that it's the first instance where the body recognizes a potential foreign antigen that needs, specific effectors for effective clearance. So you can say, or we can say that in primary immune response, no, it serves as the priming event, so that succeeding events or succeeding recognition of the same antigen, the same cell, will be faster. Okay, an antigen's route of entry is very important, no? Dictating how the initial response of the body will take place. If the antigen enters the bloodstream, the circulation, the initial response to it will happen mostly in the spleen. For those antigens or those microbes that enter via the skin or the subcutaneous tissues, we expect that the initial IR will be within the lymphatic system, lymph nodes, regional nodes, as well as the other secondary lymphoid tissues. If mucosal naman, either the gastrointestinal tract, sa no, intestines, or the respiratory tract in our mucous membranes lining our respiratory pathway, we expect that the initial IR will take place just within the locality. No? Submucosal lymphoid tissues are involved for the initial IR, the initial immune response. Where sa lahat ng mga locations na ito, what happens is, of course, we expect the antigen to be taken up and to proceed to the process of, or to proceed being processed and eventually presented. The primary cells involved are, of course, the antigen presenting ones. The antigen presenting cells we review are of different nature. Some can be phagocytes, like for the case of macrophages. We also have lymphocytes for the case of B cells. And we have dendritic cells, which are the primary antigen presenting cells for the initial or the primary immune response. So these cells that are found in non-lymphoid as well as lymphoid tissue, they circulate in our blood as well as in our lymph fluid. As I mentioned, in primary immune response, no, dendritic cells are the primary actors. So they migrate to lymph nodes, or they can also go to the spleen, where they will target or localize in T cell abundant areas. So T cells in these areas, they specifically recognize the processed antigen on the surface of the dendritic cell or the antigen presenting cell. And we expect during this time, no, yung presentation, the act of, or the event of antigen presentation will trigger the succeeding processes. No? Let's review how antigen is being processed and how it's being recognized. No? Antigen receptors are very important for the recognition of specific sensitizing antigens from the environment, from a microbial colonizing pathogen. And as we've discussed from the previous modules, we already recognize that it's the B and T cells that are responsible for recognizing antigens. No? And the recognition will be dependent on the nature of their receptors. For B cells, we have our membrane-bound immunoglobulins, our B cell receptors. For T cells, we have T cell receptors. The recognition elements of both receptors are very similar, in both B cells and T cells. No? However, the difference is that in BCRs, no, in B cell receptors, they can recognize an antigen even though it is not processed by antigen-presenting cells. Even though hindi siya associated with an MHC, a BCR can still recognize an antigen. While for TCR, they will only recognize an antigen that is fully processed by an antigen-presenting cell. An antigen that has been taken up and processed, complex with major histocompatibility complex molecules. No? Now, uh, these receptors, no, or these BCRs and TCRs, they are associated with other surface proteins. You will remember, no, yung na-discuss natin about the recognition receptor complex. So the recognition receptor complex, it's not only made up by the BCR and the TCR alone, but there are associated surface proteins proximal to these BCRs and TCRs that form association with them, and essentially they form the whole receptor complex. Okay, like for the case of our B cell receptor, they form what they call this. They form associations with other surface proteins like what do you call this? Yung alpha and beta proteins natin that are very proximal to them, assisting in the whole recognition process. Okay. Um, the antigens that are recognized by these receptor complexes, they may be of different natures. And you would remember na I've already mentioned about thymus-dependent antigens. No? Thymus-dependent antigens, one of the major types of antigens that are recognized by our immune effectors, by our adaptive immune effectors. The thymus-dependent antigens, they are recognized via the interaction of T and B cells. So they will really depend on cooperation between these lymphocytes. Okay, so your B cells will require T cell activation in order for thymus-dependent antigens to be recognized. Eventually, the process of recognizing this type of antigens will result to the formation of antibody secreting plasma cells specific to that thymus-dependent antigen, and eventually the antibody will have increasing affinity or will have increasing specificity to that antigen. Those antigens that do not require T cell activation are belonging to the thymus independent type, no? Which is further divided into two categories: type 1 antigens, T1 antigens, and type 2 antigens, T2 antigens. T1 antigens, like some examples include the lipopolysaccharides, cell wall components of bacteria. They activate B cells via toll-like receptors or TLRs. On the other hand, the second subset of thymus-independent antigens, the T2 type, include capsular polysaccharides, polymeric protein, and they activate the BCR directly via, or they activate B cells directly via binding with the B cell receptor. So yun ang difference ng T1 and T2. T1 binds or activates B cells via TLRs, while T2 binds B cells in the BCR, therefore activating them. A common characteristic of many T1 antigens is the presence of molecules that are of repetitive nature, no? Repetitive epitope. So and because of such parang ano no, molecular signature, the response to these antigens will not require further T cell help. Kasi in a way, on its own, it serves as a molecular signature already that can easily be recognized, or a molecular signature where the body already developed a specific receptor for. So yung mga ano natin, mga TLRs natin, you can say that they are more primitive sila, that they develop with us or develop through time, no? Based on the history of being exposed to specific antigens, specific T1 antigens. Okay? Now, so you know these two types of antigens, they're recognized, they're taken up and processed by our they're taken up, processed and presented such that the adaptive immune cells will develop specificity against them or specificity to them, So the humoral effectors, ating antibodies, will be specific or specific humoral effectors, specific antibodies against these antigens will be produced. Now, once that lymphocytes, eventually active, well, once that they are recognized, or once that lymphocytes, what do you call this? Nagkaroon na tayo ng antigen presentation, the lymphocyte will respond in a way, or will respond following a sequence of events. Selective binding of the antigen to specific receptors on both B and T cells is the initial step. So you expect that the receptors that are capable of recognizing the antigen will be binding to that antigen, or or the other way around. The antigen will be bound to these receptors. Once binding completes, signals will be stimulating the B cell to, or that specific event will serve as a signal that will stimulate the B cell to proliferate and differentiate into a clone of daughter cells. Those cells that, even though they possess BCR, they possess specific receptors for the antigens. Those cells that do not bind to antigens, they will remain quiescent. No? And and in contrary, those cells that bound to antigens will undergo specific activation, proliferation, and differentiation following uh, uh, antigen presentation via the MHC. No? So only lymphocytes that bind to desensitizing antigen will proceed to the succeeding events of cell division, maturation, and differentiation. No? The clonal expansion process in primary and secondary immune response, uh, quite different, no? Yung ano niya, yung sequence of events. So let's take a look. So here, let's have clonal expansion in primary and secondary immune response. Okay. So here you can see, let's say for example, now we have a hypothetical antigen X. So antigen X is presented at the start of day 0. Okay. So the B cell specific to antigen X will be able to recognize it, okay? And as such, it will differentiate into an antibody producing cell that will secrete antibodies specific for antigen X, no? Specific to antigen X. And it will produce daughter cells that are also capable of producing the antibody that is, what do you call this? That is, sorry, let me qualify my statement. So, after antigen X is recognized by specific B cells, okay, or possessing receptors that are capable of recognizing the antigen, it will initially produce a mother antibody producing cell, and ito yung it will differentiate into an antibody producing cell, not really produce but differentiate, kasi it's the first cell that will develop the capacity to produce antibodies specific to the antigen. That mother antibody producing cell against antigen X will clonally expand, or will clonally reproduce itself. Okay, and the clones are the ones responsible to produce the antibodies. Okay, so some of the clones will, or most of the clones will be very active during the peak period of primary response. They will produce antibodies against antigen X so that it can totally be cleared, while others will remain as memory cells. Eventually, there will be a downfall in the concentration of anti-X antibody. Kasi during this time, there's already an initial clearance of antigen X. No? Now, the memory cells already have the instructions for developing receptors, developing Igs against antigen X. No? Therefore, there's no need for priming to take place anymore. There's no need for priming to happen anymore. Okay, kaya if let's say in the second wave of immune response, like in another succeeding event of exposure to the same antigen, these memory cells will be easily activated, producing antibodies directly, no? Or maaari ding mag-produce pa ng succeeding memory cells. But this time, these succeeding memory cells will have higher affinity, will have higher specificity against antigen X, against the sensitizing antigen. Okay? So more antibodies will be produced, and then we expect that these antibodies can easily clear yung invading antigen natin. Okay? All right. So I guess we're now ready to discuss the molecular events, or to review the molecular events in primary immune response, especially in clonal expansion, which is very important for us to really appreciate what's really happening. So you remember that the primary area, or the descriptive term we associate to um, lymphocyte maturation, lymphocyte differentiation, is the germinal center. The germinal centers within our lymphoid tissues, they result from a subset of successful lymphocytes that already activated and clonally reproduce themselves. So these germinal centers, like, an aggregate sila, no, ng mga activated lymphocytes. And they serve as sites for further cellular differentiation, such that your lymphocytes will be very specific, no? Yung mga lymphocytes residing in these areas, differentiating, maturing in these areas, will produce memory B cells that are of higher specificity, of higher recognition capacity. So these germinal centers found in secondary lymphoid tissues, both in spleen and the lymphatic system, mga lymph nodules natin. What happens in these areas? The key events include affinity maturation, and immunoglobulin class switching, which I've mentioned from our previous modules already. The end product in germinal center reaction are cells with high affinity and cells that can easily recognize specific antigens, such as plasma cells, such as yung ano natin, no, lymphocytes natin, B cells natin, and T cells natin that have specific receptors for the antigen, are plasma cells that can secrete large volumes of high affinity antibody, and yung mga memory cells natin that are further fine-tuned for rapid reactivation for succeeding exposure to the same antigen. The germinal center processes of affinity maturation and class switching, we will cover in our succeeding discussion. So, affinity maturation is where we expect the uh, B cell receptor to improve its affinity against the activating antigen. And it involves several events, specifically starting with clonal expansion, and then diversity via, or increase in diversity carried out by somatic hypermutation, and then eventually antigen selection of B cells. Class switching has something to do with the capacity of the antibody secreting cell to switch the type of antibody it produces without really affecting the established specificity in terms of its receptors. No? And it will involve what do you call this, rearrangements, no? Gene rearrangements in the Ig coding genes. Now, let's review yung events of affinity maturation. Activated B cells, those that are T cell assisted, specifically for thymus-dependent antigens, they will undergo rapid proliferation in the germinal centers. And they assume a state of what we call clonally expanding state. No? Nagdi-divide talaga sila. We call them centroblasts. They're found on the dark zone area of our germinal center. So in this stage, no, we expect that as your antigen is bound to our to our B cell, primarily by as assisted by T cell as well, T cell activity. Okay, your B cell, activated B cell, your centroblast will continue on dividing. Now, eventually, it will undergo somatic hypermutation, where Ig coding genes will have mutations within the variable region. The type of mutation characteristic for somatic hypermutation is very limited to the variable region only. Normally, yung mga mutation na to ay base pairs lang. So, nagbabago lang ng paisa-isa. No? However, the frequency of these mutations is very high. That for each cell division, one mutation actually happens. Ibig sabihin, isipin mo na lang kung 10 times nag clonally expand ang isang centroblast, you expect that the 10th set of progeny is actually possessing 10 mutations already. Diba? We expect that B cells expressing antibody at the end of clonal expansion, no? Yung mga B cells na yon, they may either have lower or higher affinity to the specific antigen compared to the parent, because we cannot really tell whether the mutation accrued for every division will be beneficial to specificity or not. So remember that that mutations are generally a blank characteristic. It will not really tell, or it will not really have a specific or it will not really have how can I phrase it? Hindi natin masasabi that a mutation will be beneficial all throughout. No? So it's possible that a mutation accrued during a specific cell division might be harmful. No? It might lower the affinity. In some instances, it might increase the affinity of the cell towards the antigen. So there's no telling on the final effect of somatic hypermutations to the specificity and affinity of our activated lymphocytes. No? Okay. Some details about somatic hypermutation. I need to mention these things. I think I haven't really mentioned them before. No? The process of SHN is catalyzed by a specific enzyme, activation-induced cytidine deaminase, AID. What it does is it deaminates cytosine, reducing or producing uracil. So cytosine residues will become uracil in the effect of, or by the effect of the AID enzyme. Subsequently, what comes into play are DNA mutation factors, those that do mismatch repair, so that we can go back to the normal process. And usually in that, usually after mismatch repair is carried out, we expect that base pair point mutations will definitely accrue, no? Especially if, kasi ang mismatch repair system naman ng mga somatic cells it will not really be perfect. So by inducing such somatic hypermutations, eventually there is tolerable diversity or tolerable there's accrual of tolerable mutations, no, that will not really be lethal to the cell, but rather will increase the diversity and the diversity, the differences within the variable coding, the variable region uh, part of the Ig coding genes. Now, once the centroblasts stop dividing, no, they assume a state called the centrocyte state. The centrocytes migrate from the dark zone to the light zone. So here. So, diba, our centroblasts now stop dividing. They assume a centrocyte state. So the centrocyte status will not divide anymore, and it will now undergo selection. So the fittest centrocytes will be selected to proceed to the process to the succeeding events that are now confined in the light zone area of the germinal center. So B cells in the germinal center, usually they will manifest downregulated expression of survival factors such as BCL2. Therefore, they will die unless that they receive help from other cells. So these centrocytes here, they will now have downregulated survival factor expression. If they will not receive assistance from cells such as follicular dendritic cells that still possess yung mga specific antigens, or follicular helper T cells, if they will not receive assistance from these cells, our centrocytes will definitely die. Now, the assistance or the interaction between a centrocyte and a follicular dendritic cell as well as a helper T cell will depend on the ability of the centrocyte to recognize activating antigens. So it boils down to the nature of the receptor, of the B cell receptor. So, kung hindi maganda yung B cell receptor, even though there is multiple hypermutations, somatic hypermutations, if the B cell receptor is not of good quality, not of very high affinity against the antigen or to the antigen, it will not be helped or will not be assisted by follicular dendritic cells, by helper T cells. No? So, we expect that centrocytes expressing the highest affinity receptors will be the only ones assisted by FDCs, by T helper cells. Okay. Centrocytes that will be assisted by FDCs, by follicular T cells will of course receive stimulatory molecules, no, will stimulatory cytokines, and essentially they will now be transitioning to a state where they will express more anti-apoptotic genes like BCL-XL. That's an example of an anti-apoptotic gene, telling the centrocyte that it needs to survive or making the centrocyte more fit compared to others. Okay, repeated in subsequent instances in which the the centrocyte is exposed to what do you call this, to the same antigen, the sensitizing antigen, and what do you call this, pag na ano no, in the succeeding instances that it will encounter again the same antigen, usually via the APC, mga dendritic cells, as well as helper T lymphocytes specific to the antigen. Yung mga succeeding instances will fine-tune yung yung receptor ng B cell, therefore making it making the B cell more and more specific to the antigen, to the point na talagang siya lang, yung ganong klaseng B cell lang ang makakakilala sa antigen na yun. Okay? Or makakakilala ng sobrang bilis sa antigen na yon. That it's the only cell that can really bind to it in less in milliseconds or what. Okay? All right, so we're done with clonal expansion and somatic hypermutation. Now let's proceed to the succeeding step of class switching, antibody class switching. We expected the generated Ig isotype by our our plasma cells is influenced by different elements, by different factors. Specifically, the antigen composition, the route of immunization, and the nature of cytokines that are present within the locality of the germinal center where the plasma cell is maturing, where the plasma cell is differentiating. And the ability of antibody secreting cells to this ability of antibody secreting cells to switch class of antibody being produced without affecting yung kanyang specificity will involve recombination events in the DNA coding for the immunoglobulins. Usually, changes in the class of antibody expressed nangyayari siya through direct removal of intervening sequences between the constant regions. No, for example, no, IgM to IgA. Okay, from if let's say for example the specific B cell here is known to or has been programmed to secrete IgM. How can it do class switching? So the class switching can only be possible if instead of the gene coding for IgM, the gene coding for IgA will be expressed, and that process or that switch in expression will depend on enzymatic activities, usually by removing intervening sequences between constant regions of the to be developed immunoglobulin. Okay, so let's take a look at that process. In the initial phases of the primary immune response, the variable region genes are linked to the closest heavy chain gene on the chromosome. Normally, that coding for immunoglobulin M and immunoglobulin A. Kaya nga, di ba, in primary response, ang predominant na napoproduce ay IgM. Later in the process, later in the immune response waves, specifically, after re-exposure, usually followed or usually following initial clearance, if re-exposure happens, the variable region genes will become associated with other heavy chain genes farther away away of the chromosome. Kaya ang nangyayari, instead of the initial ones coding for IgM or IgA, nagkakapag-produce na ng ibang isotype like for IgG or other subtypes of IgA, no. Kaya nagkakaroon tayo ng predominance of IgG instead of IgM because instead of the IgM section being expressed, it's the IgG section that's now being expressed, no. Similar with the process of affinity selection, class switching will occur in the light zone area of the germinal center. Here we expect B-cell interactions to occur, what do you call this, to occur with the interaction between receptors and ligands. No? So CD40 in B cells will bind to ligand present in activated T cells, CD40L or GPR9 for activated T cells. Yan yung specific na ligand for our B cell receptor or our CD40. So that's an interaction between our B cell and our T cell. Then B cell proliferation takes place. Isotype switching will take place, no? Depending on, again nga, yung nature ng, nature ng antigen that is present within the area, kung ano pa, no? May specific pa ba na, or may difference ba in antigens that are being expressed, no? Then the nature of antibody class will be finally depending on cytokines that are produced. So you can think of class switching, or the essentially the production of antibody as a event, as an event that is affected by multiple factors, no, or multiple processes. Okay, cytokines that are secreted by other immune effectors, by other immune cells, will induce selective transcription of germline transcripts upstream of the relevant switch regions in heavy chain gene locus. So we expect that without necessarily, changing its receptor specificity, your surviving plasma cell will be capable of producing other Ig types, specifically of the IgG nature, without losing its affinity to recognize the antigen that is initially dependent, or initially tied with the production of IgM. Okay. All right. So now we're leading to the final stages of of the primary immune response, where you expect that um, uh, the clearance of the antigen, of the foreign antigen, and the clearance of the microbial cell, of the colonizing cell, will be the end goal. No? The decrease in sensitizing antigen will result to a decrease in the population of the specific lymphocytes, both T and B lymphocytes. No? And we expect, no, na syempre po, as time passes, the quantity or the concentration of the invading antigen or the invading cell will decrease as the body produces specific antibodies against it. And the remnants of the initial immune response are the memory cells. These memory cells will be, of course, very important, very 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 essential sila for secondary exposures, because they will no longer, or there will be no need for additional events to take place, or for the previous events of somatic hypermutation, class switching, affinity maturation, or the whole process of affinity maturation. It doesn't have to take place anymore for that specific antigen, because memory cells are already present for that specific antigen. However, the case will be different if, let's say, for example, memory cells specific for that antigen are totally wiped out. Like for the case of AIDS, no, where the memory cells specific for, the memory cells specific for the antigen, kung talagang totally wiped out sila, back to zero yung process. So kailangan mag-undergo ulit ng affinity maturation. Kaya yun ang problematic with immunodeficient patients. Ang nangyayari, the pool of memory cells that they already have, memory cells that are so fine-tuned, memory cells that are so ready to produce antibodies against the pathogen, if that pool is totally wiped out, kailangan umulit ng affinity maturation for that specific antigen. So what happens is, for immunodeficient patients, it's as if yung secondary exposure nila ay primary exposure pala ulit. Parang gano'n, no? Parang all the time it's primary exposure. Okay? So that's a problem. That's a case for immunodeficient patients. The memory cells that are so fine-tuned, so ready to produce antibodies against the antigen, against the colonizing pathogen, wala sila. Totally wiped out sila. Okay? So, now for secondary immune response, as I mentioned, characterized by memory cells that are functionally inactive if there is no exposure to a specific to the specific antigen. If we're to compare it nga, no, to their counterparts in the primary immune response, these memory cells are inactive. Okay, but they may persist in our body for as long as years. No, the survival mechanism or how these memory cells uh, uh, stay intact for a long period of time, it's still unclear. Because remember, no, from affinity selection, from affinity maturation, you would remember there should be constant supply of the antigen that's serving as a ligand to the BCR and TCR of memory cells that I mean for specific for sorry for B cells, no. Um, Kailangan may constant supply ng antigen so that the receptor, the need for expressing the receptor will be justified by the cell, because without the antigen binding to the receptor, the B cell will not be able to justify the need to express that specific receptor that is fine tuned for that antigen. So there should be a constant supply of that antigen. But of course, that's not possible in a normal or in a normal physiological state. The foreign antigen should not be present in any way, though. But what's being hypothesized is probably there is a retention of small amounts of the sensitizing antigen within these memory B cells that help them justify the need to become memory cells specific to that antigen. Now, with the subsequent exposure to the original antigen, these memory cells will be promptly activated, will be promptly stimulated, they will proliferate, differentiate, and they will produce antibodies specific to the antigen. Therefore, you have an enhanced immune response, an enhanced secondary immune response. Okay. The immune response under the secondary wave is described as increased activity in effector T cells, and the activity also, or the increase in activity also reflects to increased distribution, so change in tissue distribution. You know? The secondary antibody response will be really fast compared to the primary one. Greater amplitude at shorter periods of time. You know? And compared to primary immune response, it's more sustained, primarily because of the fact that there are already memory cells that are capable of recognizing the antigen. Memory cells that do not really have to undergo the affinity maturation process anymore. And this, process of secondary immune response is actually possessing an improved rate of neutralizing or killing capacity. Mas mabilis ang pag-neutralize or pagpatay sa invading antigen. Okay? So in a continuum, the primary and secondary waves of immune responses actually develop our rationale for immunization, no, for vaccination. Okay, that if you are to introduce a foreign material, a foreign antigen, initially you expect that memory will be developed against it. The memory B cells will be staying in the body for certain period of time. So in subsequent exposures, na kung saan, for example, no, totoong sakit na talaga, no, or totoong pathogen na yung nag-enter sa katawan ng ating animal, ng human, we expect that there are already memory cells that will respond to it. Okay? So this is a good summary of primary and secondary immune response. No? You start from your naive B cells that will be activated with the help of your T cells, your antigen-presenting cells. Okay, affinity maturation takes place. Okay, antibodies that are produced primarily are of isotype M nature. So that will cause clearance of the antigen, that will cause clearance of the invading pathogen. Provided that the antigen and the invading pathogen will decrease in concentration or will be totally cleared, there's no more need to produce specific antibodies, forming our memory cells, no? Our plasma cells. Actually, just to ano to reiterate, no, both meron tayong yung term natin na memory lymphocytes not only applicable to plasma cells, no? We also have memory lymphocytes that are of T nature, no, mga T cells natin. Pero compared to plasma cells, those memory cells that are of T lymphocyte nature, we expect them to have mas maikli yung kanilang lifespan. Because in plasma cell, the plasma cells are the ones that we really count on for succeeding for subsequent exposure to the same antigen, to the pathogen. We count on memory cells heavily. No? These memory cells, after repeat infection, now ready to produce antibodies or differentiate further, uh, so that the necessary antibodies, especially of the IgG type, will be produced. No? The, in this state, at this state, you see that there is an increased rate of antibody production, therefore increase immune response. No? At a shorter period of time. Then, ganun ulit, no? As the body eliminates the antigen and what do you call this, the microbial pathogen, we expect na some memory B cells will still be kept intact. So, kung mayroon pang tertiary or other repeat infections, the body will be ready to fight it. Okay? So that ends our session, our recorded session for primary and secondary immune response.